So for DNA replication, we already discussed earlier about the 5 different enzymes that you need to discuss, as well as at what point of the cell cycle does DNA replication take place. So just to recap, emphasize to the students that DNA replication takes place during the S phase of interphase, and then go on to talk about the 5 different enzymes that play a role in the process of DNA replication itself. It's usually easier to explain to students using some form of diagram or a video. So I've just put up a generalized diagram for DNA replication up, just to go through some of the points. So once again, to recap, DNA helicase is the first enzyme to play a role in the process of DNA replication. DNA helicase separates the parent DNA molecule into its composing 2 strands, which are drawn in the color blue. This is done by breaking the hydrogen bonds between the complementary base pairs of the 2 strands in the parent DNA molecule. The result of the action of DNA helicase is the creation of a replication fork, and we will be referring to the replication fork several times later on in the process. Now, as helicase separates the parent DNA molecule into its 2 parent strands and proceeds to the right of the board, you will see the other enzymes coming into action. The next enzyme after helicase to play a role is the enzyme RNA primase. And RNA primase produces these short fragments of RNA which are drawn in green, and these short fragments of RNA bind to the parent DNA strands drawn in blue by complementary base pairing once again, and these short RNA primers function as a place onto which the DNA polymerase III enzyme can bind and carry out the actual process of DNA replication. So once the RNA primers are laid, the main enzyme of DNA replication, which is DNA polymerase III, comes in. It's drawn in a red circle, and it carries out the process of DNA replication always in a 5' to 3' direction. Always 5' to 3' direction. Now it can be seen on the diagram I've put up that the blue parent molecule, this end represents the 3' end, which means that the new molecule to be built along this strand will have to start with the 5' and produce towards the 3', which DNA polymerase III can do without any difficulty, as it always works in a 5' to 3' direction. The result is that the new DNA molecule DNA strand drawn in black will be produced continuously with no obstacles towards the replication fork through continuous replication by using complementary base pairing with free nucleoside triphosphates found in the nucleoplasm. Now, because DNA replication takes place continuously towards the replication fork on this particular strand, this strand is referred to as the leading strand. However, on the other strand, there is a problem, because the blue molecule ends with a 5' end here. Therefore, the newly synthesized molecule would have a 3' end here. Now, DNA polymerase III cannot begin with a 3' and work inwards towards 5'. It can only start from 5' and work outwards. The result is that while DNA polymerase III works away from the replication fork, helicase would unzip further, and then this enzyme would go there and create another short fragment. This process repeats itself to produce short fragments of DNA known as Okazaki fragments. And this strand where replication happens in short bouts is known as the lagging strand. Once again, on the leading strand, the new molecule of DNA or the new strand of DNA is built without any difficulties from a 5' end to a 3' end towards the replication fork. So as helicase unzips this way, this will just follow continuously with no difficulties. But on the lagging strand, because DNA polymerase 3 cannot begin with a 3' end, it has to go into the replication fork and replicate outwards as the helicase unzips further, then proceed and copy the remaining portion. The result is short fragments of DNA known as Okazaki fragments. Okay? Now, once this much has been completed, there are two more enzymes which come into play just before replication is completed. Of the two enzymes, the first to come into play is DNA polymerase 1. Now, you will notice that there is a short primer made of RNA on the leading strand and several short primers made of RNA on the lagging strand. Now, in the final product of DNA replication, you cannot have small sections of RNA located in between a DNA molecule. Therefore, you need a special enzyme, DNA polymerase 1, to selectively come and remove these little green primers and replace them with purely DNA nucleotides. So once DNA polymerase accomplishes that task, these green areas will be replaced by DNA nucleotides. Right? Finally, you've removed the RNA nucleotides and replaced them with DNA. However, on the lagging strand, you still have separate Okazaki fragments. Now, these Okazaki fragments are connected by the formation of a covalent bond by the final enzyme to be involved, which is DNA ligase. True to its name, it ligates the separate Okazaki fragments together, forming a continuous DNA molecule even in the lagging strand. Finally, to conclude, I will emphasize to the students that the process of DNA replication is a semi-conservative process. Semi-conservative process because each of the two DNA molecules that are formed consist of one old parent DNA strand and one newly synthesized strand, partially conserving the old while partially being built anew. Therefore, this process is referred to as a semi-conservative process. So once again, stress on the fact that the process is semi-conservative, that it takes place during the synthesis phase of interphase, and then elaborate on the functions of the 5 enzymes: helicase, RNA primase, DNA polymerase 3, DNA polymerase 1, and DNA ligase, and make sure that you discuss the presence of a leading strand and a lagging strand, as well as Okazaki fragments, and how the process is caused due to the fact that replication can only happen in a 5' to 3' direction. Thank you.